MIB Agents OsteoBites webinar and podcast presents the latest in osteosarcoma treatment, research, innovation, and hope each week. Today we're talking with Dr. Chand Khanna on a comparative approach to osteosarcoma metastasis, biology, and therapy. Welcome to OsteoBites, everybody. Back from vacation today, so back to healthy eating, and so my snack is cashews, which is not that exciting. What is exciting, though, is we have an incredible guest today, Dr. Chand Khanna. Dr. Khanna is a well-known, renowned even, osteosarcoma expert and researcher. He is a board-certified diplomat of the American College of Veterinary Internal Medicine and an honorary diplomat of the American College of Veterinary Pathology. He received his PhD in Pathobiology from the University of Minnesota, and then completed a postdoctoral fellowship in Pediatric Oncology at the National Cancer Institute, where he led a laboratory focused on metastasis drug discovery. And he was also the founding director of the NCI Comparative Oncology Program. So exciting. Dr. Khanna is Chief Science Officer with Ethos Veterinary Health and President of Ethos Discovery, a nonprofit incubator for scientific innovation. Our panel today includes Amy Woodcheck, physician's assistant and childhood cancer survivor. And I'm your host, Anne Graham, executive director of MIB Agents. MIB Agents is a pediatric osteosarcoma nonprofit dedicated to making it better for our community of patients, caregivers, doctors, and researchers, with the goal of less toxic, more effective treatments and a cure for this aggressive bone cancer. MIB makes it better for kids with osteosarcoma in 3 ways: through direct patient and family support with many programs to ensure that no one walks alone through osteosarcoma. We also do this through education, including our annual Factor Conference, OsteoBites, our testing and research directory, and our book, Osteosarcoma From Our Families to Yours. And finally, through research, by funding it, sharing it, supporting researchers and physicians, and any way you can think of, we'll support research and share it. With that, Dr. Khanna, would you get us started by introducing yourself, please?
Great, thank you very much. So it's really an honor to be here, and thank you for inviting me to join this OsteoBites session. I'll share with you today, as Anne suggested, a long history for me, sort of suggesting that I'm getting quite old, of my work in osteosarcoma. And that is a history that began working with Dr. Lee Hellman at the Pediatric Oncology Branch of the NCI, working in the laboratory as a cell biologist and as a veterinarian in the field of comparative oncology, which I'll introduce today, leading me now to the work I do at Ethos, which involves the ongoing work of cross-species translation and efforts to improve health outcomes across species lines. Thank you. So if it's okay, I'll jump into a disclosure and perspective slide. And so it's certainly a disclosure of my employment within Ethos Veterinary Health and Ethos Discovery, but also disclosure of relationships that have formed my perspective and gives you a sense of why I think the way I think. And that why I think the way I think was formed as a postdoctoral fellow with Dr. Lee Hellman at the National Cancer Institute, where I did work in childhood cancer drug discovery, focused on the problem of metastasis. But during that time, as a veterinary clinician, practiced as a veterinary oncologist in the Washington, D.C. area. And that connection allowed me to then start within the National Cancer Institute, the Comparative Oncology Program, which is a field that simply says that we can answer questions in dogs that develop cancer spontaneously that we can't answer in human clinical trials, and we can't answer in conventional preclinical models. And as a result, we can deliver knowledge in ways that wasn't delivered otherwise. And that is the intersection of my background as a PhD scientist and as a veterinary oncologist. After leaving the NIH, I have now continued this work within Ethos Veterinary Health and Ethos Discovery with a focus in sarcoma and work that continues the work on metastasis as a critical problem. And that is the key financial disclosure is I am an employee of Ethos Veterinary Health and Ethos Discovery. And I'll share the work that began at the National Institute Cancer Institute, the transition to Ethos, and then share some tidbits of work that I'm involved with through Vujade Sciences and the Osteosarcoma Institute, where the recurring motif is this connection to osteosarcoma metastasis. I do have a financial disclosure related to Vujade, both as an investor and as a board member at Vujade, and as the chair of the Strategic Advisory Board of the Osteosarcoma Institute. And the Osteosarcoma Institute shares in its mission with MIB a almost singular focus to improve health outcomes for patients with this, and not surprisingly, that surrounds the problem of metastasis. And like MIB, highly collaborative in its efforts and love to find and identify ways to solve this problem together. And although I will not discuss any of the work I do within animal clinical investigation, it's a disclosure nonetheless, and it's a contract research company focused on animal health drug approval. So not related to the work that I'm going to share today, which is focused on how we can improve and accelerate opportunities for drug development in humans. This institute and this effort is focused on animal health drug approvals. So that's the introduction of my disclosures. For this audience, I can be brief. We know that long-term outcomes for patients with localized osteosarcoma has historically improved through the addition of single agent chemotherapy, and then improved again through the introduction of intensified approaches to therapy, but arguably not significantly improved in over 3 decades. And we believe that further improvements are not going to occur by simply adding more cytotoxic drugs to the mixture. And we believe that understanding the biology of this disease and the biology of cancer spread from the appendicular primary location to the secondary site, almost always in the lung, is critical for us to identify new disease targets and improve health outcomes by finding new approaches to therapy for patients. And this complexity and this biological challenge is reproduced as a result of what is delivered to us by nature in pet dogs who develop this disease naturally. As was suggested, these are my patients, and these patients have unmet needs that are similar to human patients, and there is an opportunity for us to learn about the same disease in dogs and then translate that knowledge to improve health outcomes, both for humans and for animal patients with osteosarcoma. So that's a recurring message in the work we do, is transition and translation across species and systems as a means to deliver these important advances. So this is a slide that summarizes what I think many of you have seen and recognized as the biology of metastasis in solid tumors. And I'll add at the end of the slide some animations that add some new perspectives on this problem. But this slide should emphasize one critical point, and that is the formation of the primary tumor is a wholly distinct biology from the process of cancer spread. And to this date, most cancer drug development has focused on the primary tumor, or the measurable tumor, and not the process of cancer spread, nor the lesions at the secondary site, both of which are called metastases. So that biology needs to be understood for us to expect any improvements to occur. So primary tumors occur primarily because cancer cells divide excessively and don't die. But then a subset, a very rare subset of these cells, gain the ability to move, create, or adapt the blood vessels in the body to their advantage, enter the circulation, critically resist the process of cell death when cells lose contact with substrate, evade immune detection and destruction. And then what happens is they physically just plug small vessels at secondary site. It's a passive event called arrest. The cells are, most cancer cells are very, very successful and able to make it through all of those steps, leading to the point of arrest. The cells then leave the vasculature at the secondary site. They have to do something that is very difficult, and they have to survive a hostile environment at that secondary site, where most of the cells that have made it this far actually die. And a rare subset of these cells can detect the stress that is associated with that secondary location, adapt to the stress, and then survive. And that has been the focus of our research, is how do those cells do this uncomfortable thing, which is survive, whereas most of their colonies or their, I can't think of the right word, most of their relatives are hanging to a cliff and falling off that cliff and dying. How do these rare cells survive this stress, and how do they then go on to form the lesions that are the cause of morbidity in most of our patients? And so understanding how they survive gives us insights to muddle up that survival mechanism and deliver drugs that target these vulnerable cells. And so this comes back to the idea that why not target cells that are naturally vulnerable and susceptible to death by giving them a simple nudge off the edge of the cliff, rather than targeting the cells that are robust and causing a significant problem. So using nature to our advantage to find these vulnerable periods and to target these vulnerable periods by understanding the biology and then developing new drugs. So those rare cells that survive the stress go on to form metastases, either by forming colonies within vessels or outside of vessels. And then critical to think about the fact that these metastatic lesions then metastasize again. So metastases metastasize. And the reason that's important clinically is that every one of these steps in the metastatic cascade continues to occur in a patient even after they present with metastasis. And therefore, all of these steps are biological targets that are reasonable to understand and to develop druggable approaches around. And so this process continues, and you should not be misguided to believe that any of these events have occurred prior to the patient presenting. We don't know enough to believe that that's true. And because we don't know enough and we need new approaches, we should include all of these steps as targets for biological understanding and then therapeutic advances. So a couple of the highlights: metastasis, unfortunately, is both a lesion, which is a noun, and a process, which is a verb. And that confuses people, because when I tell people I study metastasis, they conclude I'm either studying the verb or the noun, when I'm actually studying both. And we need to study both in order to make improvements in outcomes for patients. The second is a provocative thing to say, but there's some reason to believe that the secondary site, such as the lung and osteosarcoma, is actually not the first site of dissemination for tumor cells. And there's a reason to believe these cells leave the primary tumor and then go to a sanctuary site where they rest dormant for a prolonged period of time, and then they break dormancy in that sanctuary site, which may be the bone marrow. We don't know that, but it may be the bone marrow. And then they break dormancy at that sanctuary site and then colonize organs like the lung. And clinically, the reason that's important, again, is because all of these events in the cascade may occur after the patient presents with the primary tumor, or after the patient presents with metastasis, because of the last thing I said, which is metastases metastasize. And again, there's a reason to believe many of these things. I think it's short of saying there's evidence or hard data for all of these things, but I think in the context of us really needing to understand a lot more about this process, we need to be creative and open-minded to look for all opportunities. This is an overview of the talk. I've really summarized the problem of metastasis both clinically and biologically already. I will transition to discuss our work to find a protein that we believe is associated with metastasis. This is the work at the National Cancer Institute with Dr. Hellman, and how we took a cross-species approach to validate the role of this protein Ezrin in osteosarcoma metastasis, and then how we've begun to translate that knowledge into the druggable opportunity, inhibiting mTOR as a means to alter this biology conveyed by Ezrin, and more broadly recognizing that the adaptation of cancer cells to stress at the secondary site may be a valuable place to place our effort as we come to the dog and the field of comparative oncology to develop drugs to translate into the human. I'll introduce the need for iteration in the dog in comparative oncology, and then conclude with some future opportunities, which are really only loosely related by the fact that I'm interested in all of these areas, but otherwise they're thematically not related. The first is a new vision and a perspective on comparative oncology, which can be summarized by saying we are looking to develop a self-funded approach to do this research, which I think will accelerate opportunities for everybody. And we call that Comparative Oncology 2.0 or drug disco. And then the second is to look at a way to accelerate the iteration in the dog by bringing metastasectomy into the clinical practice in veterinary oncology, which is not a standard of care at this time. So that's work we're doing actively within Ethos at this time. So all of that work around the outside is related by my committed effort in my career to this work, starting with the work with Dr. Hellman, and now concluding with the work that I'm continuing to do with Dr. Hellman and others within the Osteosarcoma Institute. And all of this is aligned with this perspective that you need to be flexible and interested in moving between systems as you tackle complex biological problems and embrace that diversity of these complexities across models and across systems. So within the lab at the National Cancer Institute, we recognized that no one model or disease condition or system is sufficient to answer all questions. And models are defined by the questions that you can appropriately ask of the model, and not the other way around. So you don't add value or a judgment to any one model. You define the ability of a model to answer questions, and you answer those questions, and you move on in our setting within many mouse models to the dog with a naturally occurring disease as a means to improve health outcomes for humans with the same disease. And that is what we describe as this comparative approach. And using that approach, we began this work by subtracting differences in gene expression between a highly metastatic model of osteosarcoma we called K7M2 versus a clonally related variant, which was really non-metastatic. But it's critical to keep in mind that both of these transplantable models were indistinguishable at the cell level growing on plastic, really indistinguishable at the primary tumor level, but only differentiated based on their ability to metastasize. And so that difference in the biology of metastasis allowed us to use expression analysis. And this time, to date myself, this was the first murine 4K cDNA array. So we used that to determine what genes were differentially expressed between the high and the low metastatic cells. And we found the gene villain-2, which encodes for the protein Ezrin, to be one of the outliers. And you see the immunofluorescence staining with high expression of this protein now at the cell membrane, where Ezrin acts to connect the actin cytoskeleton to the cell membrane. And by connecting the actin cytoskeleton to the cell membrane, Ezrin physically allows the cell to engage its microenvironment, presumably sense stresses in the environment, allow adaptation and survival. And you see that high level of fluorescence staining in the highly metastatic cells, but you do not see that in the low metastatic cells. And so that was the basis for us to believe that there was something about Ezrin going on in this BALB-C mouse model of metastasis. And we did the obvious experiment, which was we took the highly metastatic cells, which resulted in metastasis to the lungs in mice when Ezrin was high, and we dialed back this expression of Ezrin. And as we did so, we reversed the metastatic phenotype in this mouse model, supportive of the idea that there's something going on with Ezrin in this mouse model. But because we could transition to a different system in the dog, we have tissue arrays where we have outcome-linked tissue array samples, and primary tumor expression of Ezrin in the dog when the expression was high led to more earlier metastasis to the lungs than in dogs that had low Ezrin expression in their primary tumor. So this was an experiment that took us literally a few weeks to do and complete. And working within the children's oncology group, were able to answer the same question over a 3-year period of time for a number of reasons that are not obstacles in the dog. But we saw the same thing showing that primary tumor expression of Ezrin in children with osteosarcoma was associated with a much more aggressive course of disease than children with low Ezrin expression. So across species now we're seeing the same thing, and this gives us the confidence to spend more time digging into this biology. And had I known this at the time, I wouldn't have sort of committed to a 20-year commitment, but that's what it turned out to be. We continued to try to understand how this single protein was having such a profound role in such a complex biology. And we took 2 approaches, and the punchline is these 2 different approaches led us to the same hypothesis. So looking at the high Ezrin expressing cells and comparing them to the low Ezrin expressing cells, Jess Cassava now did the work when she was a postdoc in our lab. And she found that there was a global difference in the function of the genes associated with the high levels of Ezrin compared to the low levels of Ezrin. And that global function was really the translation of proteins or the initiation of protein translation, which was surprising for a protein, Ezrin, that was sitting at the cell membrane, linking the actin cytoskeleton to the cell membrane. So it was a curious finding at a time, but we found the same thing when we took a proteomic approach in which we developed a list of proteins that bound to Ezrin, and we eluted off the bound proteins and resolved them by tandem mass spec. And again, from a list of proteins that we believed were binding to Ezrin by tandem mass spec, we found a disproportionate number of them. This is work of Rachel Wen in a lab. A disproportionate number of those proteins were associated with the functions of protein translation and translation initiation. So that leads to then the hypothesis that Ezrin is contributing to the metastatic cascade by modulating the biology of protein translation and the initiation of protein translation. And this led me actually back to my days in vet school, when I learned that when beef cows are going to the feedlot, they often develop pneumonia in the hours before they get to the feedlot. And the reason that is that cells can't express all proteins at all times. So when these cows were exposed to Haemophilus pneumonia, some of them would succumb to the pneumonia, and others, which had a robust cellular stress response, would adapt to the pathogen exposure and then turn on the critical proteins they needed to fight off the pneumonia in a just-in-time sort of environment. And the proteins they turn on are called weakly translated proteins. They're similar because of complexity in their cis and trans regulatory elements. So they're weakly translated by that grounds, but an active translation machinery can melt through that complexity and turn on those proteins that in that setting, the cells need to address the response to stress. And this idea has been well established in the acute phase response, like the example I give you in beef cows, certainly supported in the context of cancer, since oncogenes and tumor suppressor genes are disproportionately weakly translated proteins. And we and others would argue that in the setting of a cell that's trying to address stress and adapt to stress, that the same hypothesis and biology may be reproduced. So we extended the hypothesis to believe that this idea of adapting to stress by turning on critical proteins was critical to the metastatic phenotype, and therefore was interesting to develop as a biological target for metastasis and osteosarcoma. So we began to develop additional models to understand this process. And one of the models was to take green tumor cells, deliver them to the lung, either by orthotopic injection to the leg or by tail vein injection, and then sacrifice the mouse at a defined time period after the arrival of cells in the lung, insufflate the lung with agarose, and then make red loaf sections of the lung so that you can culture lung tissue at an air fluid interface over a long period of time and maintain the architecture of the lung in the tissue culture ex vivo environment for something like 30 days. And during that time, the architecture is maintained. And during that time, the green tumor cells are engaging the collagen microenvironment of the lung and presumably adapting to the stress associated with that microenvironment. And as we've seen in the mouse, the dog, and the human, cells with high Ezrin persist and survive and form these clusters of what we may call metastasis within this assay, whereas cells with low Ezrin arrive, but really don't survive that presumed stress. And this is interesting because this stress is occurring without blood flow in an ex vivo environment, therefore suggesting that there's something about the tumors in interaction with this hostile tumor microenvironment and the ability of the cells to adapt to that stress and survive being a critical adaptation for successful metastasis in osteosarcoma. Now, because of this assay that I've described and others that we developed in the lab, we became a laboratory of interest to many laboratories at the National Cancer Institute, interested in a number of different genes, a number of different cancers. But what we found as a recurring theme is that the arrival of cancer cells at the secondary site was a critical determinant, the arrival and survival of cells at that secondary site was a critical determinant of success and metastasis. And therefore, a recurring theme is seen across cancers and across genes presumed to be associated with metastasis. And it raises the idea that there are critical stages of vulnerability in metastasis that are alluring targets for drug development, and raises the possibility that Ezrin in sarcoma is a model to understand stress and the vulnerable response for metastatic cells. This leads us to two hypotheses and projects, and I will very briefly introduce them here, but for the sake of time, the first is that we learned that the organelle in the metastatic cell that's different from the organelle in the non-metastatic cell is the endoplasmic reticulum, and the ability of the endoplasmic reticulum to sense stress and then mobilize a response is different in high and low metastatic cells. And this work is published, so I won't go into much of the detail. I will share in more of the detail this work on the consequence of adapting to that stress. And the consequence of adapting to that stress is this idea of turning on critical proteins in a just-in-time manner to allow those proteins to allow the metastatic cells that are successful to survive and then create the problems that we see in patients. So in support of the hypothesis, we generated a synthetic weakly translated protein with a reporter construct, which is luciferase, and we deliver the construct to highly metastatic cells with high Ezrin in black. You see that they're able to melt through the complex 5' synthetic structure and report luciferase, but the cells in red, which have low Ezrin, because we suppressed Ezrin, were not able to do that. And this says that highly metastatic cells are able to turn on this complex weakly translated protein in support of this hypothesis. And it turns out that this is true across species. So anytime we take an osteosarcoma cell or a sarcoma cell, we knock down Ezrin, we knock down the ability of the cells to melt through this complex 5' UTR and express luciferase. And we use the same reporter system to image these cells in that ex vivo assay. So osteosarcoma cells get to the lung when they're metastatic and they persist, but they also turn on this reporter of this weakly translated protein in a way that non-metastatic cells do not. So this is occurring in vivo, ex vivo, and we can image and watch this occurring in these mouse models. We then went on to ask, could we develop a drug therapeutic approach based on turning on critical proteins? And we were lured to rapamycin, which is the inhibitor of mTOR, and downstream of mTOR is the process of protein translation. And we showed that in mice, we could deliver an exposure of the mTOR inhibitor that we believe would result in a biological effect. And in mice, we could turn off the translation of these weakly translated proteins during the process of these vulnerable stages in metastasis. So this is all fitting together. And when we look at the process in this ex vivo environment, believing that we are inhibiting the turn on of these critical proteins by inhibiting mTOR with rapamycin, you see that the untreated lung in this assay reveals metastatic lesions, but very, very low exposures of rapamycin completely block that process, in support of the hypothesis that rapamycin may be a drug to target this vulnerability in osteosarcoma metastasis. And these data then predicted what we had known prior to this and work from Lee's lab, that both rapamycin and the blocked ester temsirolimus both yield an anti-metastatic effect in this mouse model of osteosarcoma. So to be sure this is not just a one-off thing, we reproduced the result of rapamycin delivering a therapeutic benefit against metastasis in a number of models, both experimental and spontaneous metastasis models, either syngeneic or human cells in immunocompromised mice. And we're seeing the same trend across models, but not exactly the same result, but the same trend across model systems. And this sort of concludes the work at the National Cancer Institute, leading to the broader agenda of targeting stress, both in the endoplasmic reticulum and then the consequences by focusing on mTOR. I will not get into the endoplasmic reticulum work and focus on our effort to translate mTOR inhibition as a therapeutic approach in dogs through the comparative oncology agenda as we try to bring this forward to the human. And this cartoon summarizes what I think we all know, which is that most of us look more like our pet dogs than we do like mice. And the cartoon is supported by work from our laboratory using expression analysis to compare human and canine osteosarcoma. And I can tell you that although the colors don't show it on my slide, you can clearly distinguish normal tissues in the dog and the human from tumor tissue, but by cluster analysis, looking at thousands of orthologous genes, you can't distinguish the dog for the human osteosarcoma. And so that similarity or that indistinguishability supports the hypothesis that the dog with osteosarcoma can be useful as a means to understanding biology and therapeutic translation to the human. And although all of these are reasons that the dog with cancer and the dog with osteosarcoma lend value to translation in the human, as a cancer biologist, the text in red is most alluring to me. And that is that cell to cell to cell, the heterogeneity in the genomics of cancer in the dog is reproduced as it is in the human. And I believe that results in the essence of the problem of cancer in dogs and humans, which is recurrence, resistance, and metastasis. And as a scientific researcher, I work to reproduce that heterogeneity in mouse models, but it's very difficult to do at the best, but it's, again, delivered by nature in the dog with a naturally occurring disease. And this idea that dogs with naturally occurring cancer is not new. It began actually well before the 60s in lymphoma in dogs. And we've matured over time with a number of highlights to a point in time where we're now at, which I call the integrated period. And that is where cancer drug development, where there are many unanswered questions, should occur with a lens or a view to what questions can be answered in the dog in parallel. So we should think of this as parallel drug development, where we can answer questions in the dog that'll fuel answers in the human and then advance the field. But critical to say, during this timeline, we have now the endorsement and the support of the National Cancer Institute to do work in comparative oncology, as of the work that Lee and I did in 2003. We have regulatory guidance, both from the human and the veterinary side of the FDA, on how to do this carefully and appropriately. And we have pharma buy-in on this concept. So I think all of those lends us to take advantage of this opportunity in time. And I'd add to that the importance of having a mature canine cancer genome so we can understand why drugs are working or not working in the dog as a means to enhance translation. Taking advantage of this within the National Cancer Institute, we developed a consortium of vet schools across the US, about 22 vet schools across the US, who can then participate in prospective clinical trials and deliver results in a timely manner to inform the human. We've developed reagents and approaches to better understand the biology of response or lack of response in the dog through these trials. This consortium and this effort has been successful both from the launch of trials and seeing them down through publication. And we've appropriately advocated for how to do this work in academia, in pharma, and most recently with the buy-in from regulatory bodies. With a meeting of many of the people that lead this field, we asked, how is it that we now develop drugs for this biology that's wholly different than primary tumor development, when we understand that drug development in pharma classically targets proliferation and apoptosis, but ignores all of the steps involved in the metastatic cascade? How do we do this for the unmet need in childhood osteosarcoma? And how do we do that at the preclinical level? And how do we translate that into the human? So this paper summarizes it, but I can tell you that a large part of it was taking advantage of the dog with osteosarcoma, building on the buy-in from this group of experts on the opportunity to ask questions in the dog. And important to say that we know that drugs that are active against these vulnerable metastatic cells and therefore have the promise to deliver value in human patients with this critical unmet need, these drugs which are successful in this setting are not successful in measurable lesions, which are typically the lesions that are measured and evaluated in early phase human clinical trials. And we know the COG has taken a big step from moving to a creative design of phase 2 osteosarcoma trials to address this challenge, but it tells you that conventional drug development is not going to find these drugs, and we need to do something different. And that continues to be sort of the mantra that I work around. The specifics from that paper were really understanding the targets and the biology of these disease targets associated with these vulnerable metastatic cells, doing robust modeling in vivo, ex vivo, and through large animal studies, and critically understanding, when you can, the pharmacokinetics and pharmacodynamics in the disease state. A lesson that I'll come back to later in the talk. This idea has been propelled, this idea of the dog as a model is propelled by the fact that there are something between 10 and 100 times more dogs with osteosarcoma diagnosed every year than children. And that prevalence of the disease in the dogs allows us in the veterinary world to conduct trials and answer questions in a very facile manner and deliver those answers to the human such that the clinical trial that's decided on for the human can be most likely to be successful. And it's important to say that we don't want to use the dog to only say is a drug active or not, but to help prioritize drugs that are under consideration in the human and pick the winner for those rare upfront human clinical trials. And this builds on the idea of us hypothesizing that mTOR inhibition with the rapalog or with rapalogs or rapamycin may be an attractive hypothesis or idea. We've had buy-in from a number of invested collaborators, including the Morris Animal Foundation, a leading animal health philanthropic group, the Quad W Foundation, to partner across species lines with what's called the Osteosarcoma Project now, and work continued by Dr. Amy LeBlanc, who directs the Comparative Oncology Program at the NCI. So this program seeks to then accelerate this process of osteosarcoma translation from the dog to the human. And you can see that the Morris Animal Foundation and the Quad W Foundation in this setting are working together to build a marketing presence around this opportunity and critical work that is ongoing outside of my purview now by Dr. Amy LeBlanc. As I think about the translation, there are many steps. And so we have built a biological rationale for rapamycin's use in metastasis from the work in Ezrin. We've explored the validation of this promise in a number of preclinical models, and we've begun to do the same thing in the dog, doing PK and PD studies in the dog with this drug. But it's important to know, as an anti-cancer drug, we don't know what the optimal PK is for rapamycin, and I'm sort of tipping my hat to later results from the discussion, but that we know about its use as a immunosuppressive drug, and that may or may not be relevant to its use as an ideal anti-cancer drug, or more specifically an anti-metastatic drug. But these studies have been done, they've been published, and I won't go into them in more detail. We then went on to really model the PK, PD, looking at the PK modeling in dogs based on the immunosuppressive history of the drug rapamycin, and understood whether the drug could be used in a chronic setting in dogs, leading to a randomized prospective study where we would have dogs receive amputation of their tumor-bearing limb and then be randomized to standard of care chemotherapy alone or standard of care chemotherapy and rapamycin. And I'm giving you the hint by the next step being the topic of iteration in comparative oncology. The hint is it didn't work. And that's disappointing on one level, but maybe rewarding, because reproducing this exact study design in the human would be a mistake. So this says that adding rapamycin to standard of care chemotherapy in the minimal residual disease setting does not deliver a survival advantage to dogs with osteosarcoma. And this circle represents the exposure to rapamycin. And that's important to know, because obviously the process of metastasis and all of the steps in the metastatic cascade are going on through the course of the disease. But in the study, we only exposed the dogs for a short period of time. Part of that was related to tolerability. And so you could ask the question, what's next for osteosarcoma and rapamycin? I think the short answer is we need a better rapamycin formulation aligned with its use as an anti-metastatic drug. We need earlier exposures, maybe in the neoadjuvant setting. We need longer exposures through the course of the metastatic cascade. And we could then dig into the question that although there's a no-sum gain here, that there may be responders and non-responders that we can decipher by reanalysis of these dogs, both at the genomic and proteomic levels. And that's sort of captured in the topic of selectivization, which are all of these are part of what is ongoing within the hands of a number of unaligned groups. We may also conclude that we were wrong and that we need to move on from this drug to others. I think all are fair ideas. And as we go on to the future opportunities, you'll see where I think some of the iteration may occur. And all of these are unrelated efforts, but they're connected by my interest and involvement in the field of metastasis. So the first is, can we shorten the wheel of iteration from the dog study that I summarized, which was over 2 years of work? And one of the ways we can do that is by introducing minimally invasive surgery and metastasectomy to dogs with osteosarcoma. So we could do trials in dogs with osteosarcoma that are more similar to the current phase 2 trials in children, and likely then add new therapeutics and deliver a more rapid result, which would be reflective of its metastatic, anti-metastatic activity. Dogs don't recover well from a median sternotomy. And so this is one of the reasons that metastasectomy, although used, has not been widely used in dogs. So if we can shorten the recovery time through a minimally invasive approach, we'll be able to add innovation and iteration to this work. And so this is part of the new work that I'm involved in dogs with osteosarcoma through my work with Ethos Discovery and its collaboration and planned collaborations with the Osteosarcoma Institute. If I more broadly take a step back and say, where are we in the field of comparative oncology? We're on a long road that we're on, at least the road, but there's a long road ahead. We have a national capacity to do prospective trials. We have regulatory clarity, a mature genome and reagents to work with. We have endorsements from the NIH, from the Institute of Medicine, from the FDA, and specifically CDER around de-risking the risk that pharma would perceive they have about doing a veterinary study while they're developing a drug for breast cancer or what have you. There's been a statement in public on the record by CDER to de-risk that concern that exists in pharma. And there are comparative oncology programs within pharma groups and certainly awareness within cooperative groups aligned with osteosarcoma, namely the COG and SARC. And so you could say with all of that progress, we just need more chances and we just do the same thing and eventually we'll get a home run. Or we can try to do things in a different way. And one of those is to more closely align the commercial interests of animal health and human health. Thus far, there have been academic alignment, but not commercial. And second, we can align CMC on the animal health approval side with the human health approval side to reduce the cost associated with moving between species and therefore create a self-funding model. And the self-funded model is simple. This is the figure from the paper. Basically, human pharma develops drugs and at some point in the development may exit financially by licensing a drug that looks like it has potential promise to a veterinary company. In exchange for that license, the human company gets data and knowledge. Sorry, I said that incorrectly. The veterinary group and the human group get data and knowledge about the use in their relevant species. The human group gets dollars from the licensing agreement. The human group gets dollars to do more research, and therefore you have a self-funded prophecy of parallel drug development. And this is what we call comparative oncology 2.0, which is part of what we're advancing within my work at Ethos now. And I'll go on to discuss the ongoing work with the topic of metastatic endurance and the phenotypic screening that is expanded from the work I did at the NCI to the work that's being done at Vujade. So I've disclosed my financial connections to Vujade, but Vujade, as you know from presentations and their involvement with MIB, actively and focused and critically focused on the problem of metastasis. And very briefly, Vujade has added to the one assay that I've focused on in the work we've shared so far, 2 different 3D assays that collectively focus on the vulnerability of cells arriving at a secondary site, cells adapting to that stress and then surviving. So there now is a panel of assays held within Vujade to find drugs that'll then be ranked and then developed based on their feasibility for development, but we'll come back to the possibility of answering questions in the dog on the way to the human. And that's all work that David Warshawski and my friends at Vujade are doing. And I won't say anything more for the sake of avoiding any disclosures that would be inappropriate, but I encourage you to go to their website to learn more. So that's where the work on targeting these vulnerable cells continues after my exit from the laboratory. And then last one is going to be then to return to my work within the Osteosarcoma Institute, which continues this theme broadly, but has a number of important opportunities, most importantly for you to hear active interest in all forms of collaboration. So any collaboration interest you have, please reach us within the Osteosarcoma Institute. And as I said, I chair the Scientific Advisory Board within the Osteosarcoma Institute. There is a hotline that's available to connect patients to experts in the field and help them in their journey with their current oncologist and other oncologists and other opportunities. There's an active effort to raise funds and find partners to do research, clinical research, correlative science, which is a critical interest of mine, which basically says, let's do more biology discovery, more work in the human with the disease so that we can iterate and make advances more quickly than we have so far. A sweet spot for the Osteosarcoma Institute, which is to fund translational and basic biology research. And the key points are that the Osteosarcoma Institute's mission is to improve survival for patients with this disease. And the second most important point is that all forms of collaboration and innovation are encouraged. Acknowledging the National Cancer Institute, Dr. Hellman, for his mentorship and friendship for now what's probably close to 30 years. Dr. Amy LeBlanc, who leads the Comparative Oncology Program, and to all of the people, and they could fill this entire page, postdocs and staff scientists within my lab at the NIH, who led us to this interest in these vulnerable metastatic cells. And lastly, to the team that I work closely with within Ethos Veterinary Health and Ethos Discovery. And I encourage you to reach out to us as well if you have ideas, your own ideas that you think we can help through this interest in moving across systems and species.
Thank you, Dr. Khanna. We at MIB are aligned in our mission and in our values with you and with Osteosarcoma Institute. In all we care about is let's get to a cure, let's get to kinder treatments minimally. Don't really care about credit. Let's just go and collaborate. And I think I always say this, but really, really relevant, that there's never been a better time for discovery. I don't know of any other patient community who's so connected, so involved, and so collaborative as the osteosarcoma community. We have to get to a cure. Like we just have to do better. It's too many kids are dying and people are dying, and it's just, I've had it. Like we've got to do better. Okay, so we have quite a few questions. One of them is this is kind of an off question, but Dr. Khanna, you're the man to answer it, I think. There's anecdotal talk always in the osteosarcoma community about who has psoriasis. And there seems to be anecdotal again, but a high correlation of kids who have any kind of a skin problem, specifically psoriasis, that sort of translates to having osteosarcoma later. Like one seems to always predispose or precede at a minimum a child with osteosarcoma. Have you seen this in canine patients? Have you heard of this phenomenon?
I haven't heard of it before, and I can say that I've not observed it in dogs. Dogs don't, get psoriasis per se, but they certainly get immune disease of the skin, and it's not an association that I'm aware of. I mean, there are very few risk factors in the dog, trauma, bone infarcts, bone implants are known to be risk factors, but I'm not aware of a immune disease of the skin. But I will share one connection to the highly interesting and fashionable topic of, immunotherapy. And that is that dogs that have infection at their limb salvage site have the longest reported survival in osteosarcoma compared to anything else. So something about stimulating the immune response is one hypothesis that I allure to, and that infection sets up, a natural Cooley's toxin. And so I think there's certainly something there. And despite the fact that we've missed, or I shouldn't say missed, despite the fact that we've not seen the immune checkpoint inhibitors deliver a big jump in benefit in patients with osteosarcoma, I have a strong belief, and a good friend of mine, the late Isaiah Fiddler, said, when you believe something, it means you don't have data. And so I believe that there's something there, but I don't have any data beyond these anecdotes that I've shared.
Yeah.
So I wonder if the psoriasis connection, could have a connection to, but as a risk factor, I really would just be safe to say I don't know, and I've not heard of that.
This one might be a little bit outside of what we're talking about, but what tests should be done on a lung metastasis? How do we get more drug trials into canine patient trials? How does that happen? Like, how does that work?
Well, we're trying to address the funding side by, as I said, trying to create a financial incentive for human and animal health pharma to do this. And so far, we've been doing this in an academic setting, and there are now 2 good examples of veterinary drugs that have been approved and are generating revenues for animal health companies. And those companies bought the rights from human companies. And so therefore the human companies acquired or were delivered revenues and returns at a relatively low risk profile. So I think that when we can convince people of the commercial value to do this, more of this will be done because we'll have erased the funding side. As I said, I think we need to, on the veterinary side, deliver results in a timeline that is actionable on the human. We can't take 2 or 3 years to say, I have a great idea and I'll tell you what happens in 2 or 3 years, because a lot's going to change in 2 or 3 years. So we have to fix that on the animal health side. And that doesn't speak necessarily to more, but when we get the metastasectomy trials figured out, I think we'll be able to do more of those. But I want to sort of share a caution. There's a lot of cottage industry that's going into this. So to do a metastasectomy in dogs, we need to 3D print the tubes to do 1 lung ventilation. These are devices that you buy for human patients. We need to print them from scratch.
Wow.
So there's a lot of work we have to do to get to a point that we can deliver on this. But as you've seen, a lot of time, a lot of work hasn't stopped us before. So we're excited about taking that on.
Right. Okay. Well, we've got to wrap it up. We're in overtime, so I'll finish by letting you know what you need to know from MIB agents this month. Is that our osteosarcoma resource packets are ready. This is a packet that contains the book that we talked about already, that's available in Chinese and Spanish and English. It's called Osteosarcoma from Our Families to Yours. There's resources in the packet from our partners at Osteosarcoma Institute and the Amputee Coalition, and a little treat or 2 as well. They're available to osteosarcoma families and to institutions who serve osteosarcoma patients. Please go to our website and request this valuable resource. Really need to get these into the hands of patient families. Also, April is jam-packed with osteosarcoma stars, including, of course, Dr. Chan Khanna. Next week, we have Dr. Jane Yanagawa, a thoracic surgeon, followed by Dr. Sumit Gupta, Dr. Inez Lose, and Dr. Dan Prince. It's 5 in April. We're like killing it in April here with really amazing speakers. Thank you for joining us today. And of course, thanks so much to our guest, Dr. Khanna. Really an honor to have you, and such interesting and compelling information. Thanks for your work and for your dedication to osteosarcoma patients everywhere.
Thank you. It's a pleasure.
Thanks to our panelists, Amy Woodcheck, physician's assistant. And thank you for being here with us. This will be on our YouTube channel and anywhere you get your podcasts. So thanks, everybody, and we'll see you next week.
Thank you. Bye-bye.
Be sure and subscribe to our YouTube channel, where you can view our library of this and all OsteoBites topics and rockstar speakers. You can also listen to OsteoBites via podcast wherever you get your podcast.